Good evening, Vihan. Vihan? Vihan, am I audible?
Yes, ma'am.
Yes, good evening. So, where are your friends? None of them are here.
Ma'am, I don't know.
That's okay. You're not here to sit with the friends. You're here to learn, right? That's okay. That's absolutely okay. If they're not there, let's learn ourselves. So, last class, everything you understood?
Yes, ma'am.
Okay. So, let's take up today replication. So I think I stopped where when replication I was supposed to begin, right? Did I stop there? Vihan? Did we finish?
Yes, ma'am.
Did we finish RNA also? RNA world?
No, ma'am.
Okay. So we didn't do RNA world. Okay. You are from RNR?
Yes, ma'am.
Then Arya also should be there, right? To the class. So what happened to him?
I'm not sure.
Indranagar students have UT tomorrow. I understand that. Okay. Let's go ahead with this. That is DNA replication. So till where we learned in the previous class. Did I write all those? Okay. Okay. So what I do is I'll explain everything and I'll send you the notes, different notes. Is it okay?
Yes, ma'am.
Yeah, I'll send you the different notes. Okay, so that should be better. I'm just opening the page. Okay. So you can see the screen, right? Vihan?
Yes, ma'am.
Okay. So who else have joined? Nobody else, no? No, none of them have joined, only we both, fine. So replication, what is replication? So let's understand replication first before going to RNA. So what is replication? When this replication would occur? What is replication? So replicate the copy. So what do you understand by that word? Replication means what?
Ma'am, it's basically copying something and
So
producing.
can we
something.
say it as duplication?
Yes, ma'am.
Yeah, replication is nothing but duplication. Okay. So it requires copying from a template. It requires copying from a template. Right? So why is it required? When does it happen? So last year you have learned this. Why is it required and when does it happen? Can you tell me? Vihan? There's no option for you. You have to answer. Vihan?
Yes, ma'am.
Yeah, when duplication would happen? Which, when it happens? During?
And when we have to create another pair of chromosomes.
When another pair of chromosome is created? When does it create? When is it created? When is it created? When is it required? DNA copying, the word, I'm using it. When is it required? Whole first unit, this is what we learned. During cell division. So when duplication would occur? Chromosome duplicates. So this is the chromosome, and this chromosome will have two chromatids, which we call sister chromatids. When does it happen? This becomes like this. When? In S phase. So that means it is replicated. So this replication is nothing but duplication. So Watson and Crick, who proposed the double helix model, immediately proposed a scheme of DNA replication the moment they proposed the model, that is in 1953. So what is that they suggested? The two strands would separate and act as template for DNA synthesis. DNA is a double helical structure. The two strands would separate and act as a complementary strands. So now, after the replication, each strand, so that means two strands would separate. Okay. Okay. So this is how. So this is the old strand, and this will become the new strand. So this is old, and this will become the new strand. Are you understanding?
Yes,
Vihan?
ma'am.
Yeah. So and act as a template for the synthesis of complementary strand. So after this completion of replication, each DNA molecule will have one parental strand and one newly synthesized. One parental strand, one newly synthesized. Therefore, we can call it as this scheme as semi-conservative DNA replication. This is told by Watson and Crick. But how far it's true? So someone has to experimentally prove it. So that's done by Messelson and Stahl. So in your textbook, they have given all about Messelson and Stahl. So Matthew Messelson and Franklin Stahl. So they perform the experiment, isotope tracer experiment. So I've already told you several times, isotope tracer experiment is the most significant experiment one can perform, because it is almost 100%, it is evident and true. So they used nitrogen isotope. So what is the nitrogen in nature? This is a heavy nitrogen. Which nitrogen is available in nature? What is the atomic mass of nitrogen? Which one is abundantly available in nature? This is the isotope. Which one is abundantly available? N14 is abundantly available. Okay, so this is available in E. coli. I'm sorry. This is available in nature. So Messel and Stahl worked on E. coli, Escherichia coli bacteria. What they did? So they grew E. coli in a medium containing heavy isotope ammonium chloride. So as the only nitrogen source for many generations he used. So it's a heavy isotope of nitrogen, and it can be separated from the regular nitrogen, that is N14, by density gradient centrifugation. So if you do the density gradient and centrifugation, so it can be separated because it is a heavy amount. It's not a very crucial radioisotope. Are you understanding, Vihan?
Yes, ma'am.
Yeah. So please keep responding, so that otherwise I would feel that you also have left the meeting or what. I would not even come to know. Okay. So this is a heavy isotope. They use this ammonium chloride to grow. This was incorporated into the newly synthesized DNA as well as other nitrogen. So what is the experiment? So you said that they are doing this particular experiment, this particular lesson in your school. So what is that experiment all about? Can you tell me? What's that experiment? Messelson and Stahl. So they did it in E. coli at the same time. So that is in the same year. So other two scientists, that is Taylor. Okay, so Taylor and et al., that is others. They did it on Vicia faba. Vicia faba is a leguminosae family, long beans. Okay, so Taylor used, instead of nitrogen, tritiated thymidine. So that is tritium. This is a heavy hydrogen. So these people used nitrogen. So what they did, they grew E. coli in a medium containing heavy nitrogen ammonium chloride. Are you understanding? So the result was N15 was incorporated into newly synthesized DNA. So what is that they did? So this is what they did. Okay, so what is the nitrogen here? Can you tell me first time? So the nitrogen, so these two are divided, E. coli divides within 20 minutes. So after 20 minutes, they got two uh, strands of DNA, double stranded DNA. So what is that they noticed? What's that they noticed? Vihan. So N15 was incorporated into newly synthesized DNA. What happened? The heavy DNA molecule, where did they distinguish it? How they could distinguish that heavy DNA molecule? So they centrifugate this in the cesium chloride density gradient. So they centrifugate this in cesium chloride density gradient. Centrifugation they did. So what happened when they centrifuged it? So what is that they noticed? Can you tell me? So both of them. So this remained N14. So this remained N14 and this became N15. So that's what they did. So N15 they grew. What happened after another 20 minutes? Okay, so what is that they noticed? So 20 and that also. So they noticed 4 different DNA strand. So how did they figure out this DNA strand? So this red acted as template and produced one strand, and red acted as template and produced the strand, and N15. Okay, so after the second, so N15 also acted as, and they produced the strand. Are you understanding? So this is
Yes, ma'am.
N15 and this is N15, this is N14. Okay, so what is that? How did they get? See, if you see the centrifugation tube. So if I draw the centrifugation tube, so let's say that this is the centrifugation tube. So in this tube, so here there is a N15 gradient. So N15 they give. So both of them N15. So this is a heavy. So what is that they got in between? In between they got the hybrid. So this is N15 and N15 and N14. So they got hybrid. Okay. So now in the third setup, so this is N14, N15. So here both of them will be N14, N14. So that is centrifugation during centrifugation in the cesium chloride. So this came to the top and this remained in the bottom, which is a hybrid, and this is a lightweight. So this showed that, this proved that. So what is that it proved? What's that it proved? Replication of DNA is a semi-conservative mode. Are you understanding? Is that clear? Replication of DNA is a semi-conservative mode. That's what they proved by this particular experiment. Are you did you get it? Vihan?
Yes, ma'am.
Okay. So what is that mechanism then? The mechanism of replication, how it happens. So let us understand that little more in a brief way. Okay. So mechanism of replication. So this particular this one, I'm just using it as teaching. So I'm not sending these annotated notes, just using it as a board and teach you. And I will send you the other notes which I have done already in previously for See, here I have done. I'll send you this. Okay. Is that clear? Vihan.
Yes, ma'am.
Yeah, neatly I have written here because you are alone. So I'm just using it as a board, like to explain, randomly writing all these. Okay. So now, DNA is a double strand. So it has to get replicated. So that means each strand has to get separated first. Do you agree? Double strand has to get separated and then only it would get replicated. Do you agree? Yeah? Are you understanding?
Yes, ma'am.
Yeah. So there are so many enzymes involved in this replication. So the main enzyme that is involved in replication is, always keep in mind, DNA dependent DNA polymerase. Polymerization of DNA happens by DNA only. So that's why DNA polymerase. Why do you use that name? Why do you use that name? Because this uses the DNA template for the DNA polymerization. That's why DNA dependent. If RNA dependent DNA polymerase, then we call it as reverse, isn't it? So these enzymes are highly efficient and they involve in the polymerization of a, what do you mean by polymerization? Which is the monomer of DNA? Which is the monomer of DNA? Quickly. So if I ask you what is the monomer of DNA, what should be your answer?
Ma'am, nucleotide.
Nucleotide. Exactly. The monomer of DNA is nucleotide. Okay. So this nucleotide, let's say that each nucleotide. So pearl mala. So each pearl, when you make a string, it becomes a polymer. This is what has to happen. Is it that easy? So threading the pearl is an easy one. So how is it here? So these enzymes are required. Many such enzymes are required. If we consider E. coli, so if I take E. coli bacteria, so E. coli has 4.6 into 10 to the power of 6 base pair. So compared to human, and it is a haploid set. So what about human beings? Compared to E. coli, how many human beings? So in human beings, it is diploid set, and it's about 6.6 into 10 to the power of 9 base pairs. This completes the process of replication. So this requires about 18 minutes for the average. On the whole, 38 minutes. Average, 18 minutes. So that's what is given. Average, 18. Average means by 2. Okay. See, suppose any mistake happens during replication, will lead to chromosomal aberration, mutation, which is nothing but mutation. So therefore, replication is an expensive process. It's very expensive. It costs a lot of energy. So now we are talking about, there are two cells. One is prokaryote, another is eukaryote. So is the replication same in both? No. So replication in eukaryote occurs in the S phase of the cell cycle. So it is semi-discontinuous. That means it's not totally discontinuous, semi-discontinuous. That means one strand is continuous, another strand is discontinuous. That's called as semi-discontinuous process. Please keep this in memory. How about prokaryotes? So in prokaryotes, replication occurs in the cytoplasm. So this is the first rule. In eukaryotes, it occurs in nucleus. In prokaryotes, it occurs in cytoplasm. Make a note of it. Is it clear, Vihan?
Yes, ma'am.
Yeah, so in eukaryotes, it occurs in S phase of the cell cycle. So how about in prokaryotes? When do you think it's going to happen? Okay, to understand that, what is the process of division in prokaryotes? How do they divide? How the cell divides? There are so many phases in eukaryotes: prophase, metaphase, anaphase. So that is cell cycle interphase. In the interphase, you have G1S, G2, then M phase. When it comes to M phase, it is prophase, anaphase, metaphase, telophase. So there are so many phases. So how about the replication in prokaryotes? What is the mode of cell division in prokaryotes? Can you tell me? Replication takes place in cytoplasm just prior to fission. Just prior to fission, replication takes place in cytoplasm. So it's a complex multi-step process that require lots of energy, lots of enzymes, protein factors, metal ions. Okay, so many things. Apart from enzymes, protein factors and metal ions are cofactors. So all these are required. You have learned all those in biomolecules last year. So how is this replication? Let us understand one by one. So both prokaryote, eukaryote. So how is it? So we are learning here more with prokaryotes because in eukaryotes, it's a very complex process. Okay. So now, very first step in replication is origin of replication. How replication occurs? So the DNA strand, which is double-stranded, somewhere the replication, the copying should begin somewhere, right? So that area, that point where the copying begins, and that is called as origin of replication. And this is called as ori. So bacterial and viral DNA has a single origin of replication. Viral means once they enter the host body, not when it is outside. Outside, it is simply a crystal molecule, crystal, which cannot replicate. So that's why we consider that as just a molecule, not a organism. No, acellular, right? Are you understanding? Vihan? Vihan? Oh, Amrita, Arya, Vihan, all three of you are there. Now that's nice. So Vihan, why are you not talking? Vihan, because I am teaching you, I thought only you are there. Vihan, can you please speak? Arya? Arya? Arya, can you speak up? I'm just testing my speaker. Amrita? None of you were talking. Am I audible? Am I audible? Guys, am I audible? Unless you talk, I cannot proceed. Vihan, Amrita, Arya. Are you understanding whatever I am teaching? Am I audible? What happened to Vihan all of a sudden? Vihan, am I audible?
Ma'am, can you hear me?
I can hear you. Was I audible before 2-3 minutes ago?
Ma'am, I had internet issues.
Then why, please, I'm not going to get to see the chat because I'm using iPad today. I'm not using laptop. Kindly speak up. See, I'm not sharing the screen. Now it is shown, but when I share the screen, it's not shown. Please speak up. Okay, so we are talking about this. So bacteria and viral DNA has a single origin. Underline and keep it. Are you understanding? And this single origin or single replicating unit is called as replicon. That unit, so single origin. So for example, so if the bacteria, so if this is the double strand, so now origin means what? It forms a loop. So this is how it forms a loop. So now from here unwinding starts, and this unit is called as replicon. Okay, so in bacteria it is single unit and replicon. In eukaryotic DNA there are several units. So if there are DNA strand, it might open here. So now it might open here. So there are so many replicons, multi-repliconic. DNA replicons are many in eukaryotes. In prokaryotes, it's single. This much is the second thing. Are you getting my point? Is it clear? This much, is it clear, guys? One of you say yes. Vihan, you were telling me, please continue, dear. Say yes, no, something. Vihan. Vihan, did you understand?
Yes, ma'am.
Yes. So point number one is what is a step one? Stepwise I'm teaching you. Replication takes place in a stepwise manner. Step one is origin of replication. Origin of replication. Step two. What happens in step two? Activation. Activation of deoxyribonucleotides. We are talking about DNA. That's why deoxyribonucleotides. Activation of. What do you mean by activation? See, these deoxyribonucleotides, which are those deoxyribonucleotides? Can you tell me? Anyone? So let's say that this is the cell. Understand, I'll send you the notes. Do not worry. But understand now, this is more of understanding than knowing the notes. So now this is your nucleus. Okay, so this is the nucleus. So now when I say activation, what do you understand by that word? So these deoxyribonucleotides are present freely inside the nucleoplasm. So they are present freely. So that is deoxyribo AMP. What is AMP? Deoxyribo AMP means what? Adenosine monophosphate, guanosine monophosphate. Then quickly go on. Then you should be power packed class. One student or 10 students, if you are there in your mind, if you know the concept, go on telling me what is the next. Arya, I don't want to prompt you children, you should be telling. This is all in the previous class we learned. What's the next one? Vihan, go ahead dear, I'm very happy about your responding the way. Deoxycytidine monophosphate and deoxyuridine monophosphate. Am I right? Am I right? Am I right? All three of you, please unmute and say, am I right? Vihan, am I right?
Ma'am, I'm not sure.
Hey, how can you say not sure? How can you say not sure? You know it. I am very sure that you know it. So is there adenine in DNA? Okay, what are the purines and pyrimidines present in DNA? Give me. What are the purines and pyrimidines present in DNA, dear? Oh. Which are the purines and pyrimidines in DNA? Vihan. We did that in the previous class. Which are the purines and pyrimidines, my dear?
Adenine, guanine, cytosine, thymine.
Exactly. So cytosine and thymine. What is that I have written here? What's that I have written here? I have written urine, uracil. So uracil cannot be uridine. So it is thiamidine. So I should be writing DTMP. Are you understanding? Instead of T. So this is what you should take. So these are all present in the nucleoplasm because they are in monophosphate. So now activation should happen. So what is that activation means? They are first phosphorylated and changed to active form. How do they get phosphorylated? So the phosphorylated, how do they get phosphorylated? The phosphorylated form will be AMP becomes ATP, DATP. So this becomes DGTP. This becomes DCTP. This becomes DTTP. So how simply it becomes like that? How do they get 3 phosphate residues instead of 1? So energy and phosphorylase enzyme come to the picture here. So energy, again, ATP itself is energy for the formation of that. And phosphorylase enzyme, phosphorylase. Is the enzyme. So they attach with H3PO4, that is sulfuric acid, sorry, phosphoric acid. So H3PO4 along with that, so they get transformed. So that is activation. This is called as activation. So in addition to acting as substrate, they provide energy. So not only they are activated deoxyribonucleotides, they are also the energy precursors for polymerization. Is that clear? So this is the step 2 process in replication. So now let's go to step 3. What is that? Exposure of DNA strand. exposure of parent DNA strand. So now DNA duplication has to happen. So exposure of the strand has to happen. So how this strand would get exposed? Why do you think the strand would get exposed? So that is DNA strand is like this, right? So now this strand should become exposed, means unwinding should happen. So how this unwinding happens? Which enzyme? Here comes the first enzyme, enzyme helicase. This enzyme, in some books, they give it as unwindase. Please learn. So this is called as unwind. Just a minute, students. So this is also called as unwindase. Enzyme helicase and unwindase. So they act on the origin. They act on the replicon or origin. So that is replication. Are you understanding? So what do they do there? How do they act on it? So unwinding, how do they bring in unwinding? So what is between the two bond here? Nitrogen base. Which bond is between the two nitrogen base here? Which bond is there, students? This is all very easy, no? Which bond is there? Which bond is there? Guys, Amrita, which bond is there, dear? So now here, 2 DNA strand is, what is there here? Which bond is there inside here? There are 3 bonds, of course. So phosphodiester bond, then peptidal bond. So which bond is there here? Glycosidic bond between the nitrogen base and the sugar, and between the 2 nitrogen base, which bond is there here? Guys, which bond is there here? Oh, Vihan. Arya? Okay, I'm not able to see the chat, students. This is very demotivating if you're not giving me which bond is there. This is hydrogen bond between the nitrogen bases. So when the hydrogen bond breaks, unwinding of DNA takes place. So this results in, so if we take unwinding, if we cut this, so it results in Y shape, fork-like structure. So now we shall go with the schematic representation of this fork. So now how it looks like, so this is like this, and this is like this. So this is like this, and this is like this. Okay, so it happens like this. So this is called as replication fork. Is it this much clear? Vihan, I think I'm teaching, I feel like I'm teaching only Vihan now. Vihan, are you understanding? Is this much clear, dear? Vihan? Vihan?
Yes, ma'am.
Oh, please answer. Is it clear this much? So now, if you take an electric thread or that cable or so wherever winding up is there like this. So the moment you unwind and release it, what have you seen due to the tension? So they again tend to wind up. So that tension has to be released. That so therefore the exposed single strand requires stabilization, and that stabilization is brought by single stranded binding proteins. So the first enzyme is helicase, which helps in unwinding, and the second enzyme is single stranded binding protein, which helps to stabilize. Is it clear? So, is that clear? Guys, are you understanding or not? One second, let me show you that picture. I have the picture, beautiful picture. Let me show you that. Yeah, can you see the picture now? Guys? Can you see the picture? Are you understanding? Can you see the picture? Very fast is unwinding, unwind days. So what is the other name I have given for you? Unwind days and, oh, it has gone so up. Helicase. So this is the first one. So stabilizing protein, single stranded binding protein, which stabilizes. So next is activation. So it breaks the hydrogen bond here. Okay. So the moment hydrogen bond is broken, so the replication fork occurs. So the exposed single strands are stabilized here by single stranded binding protein. That happens. So then, because of this unwinding, what is happening? So a supercoiling of the single strand takes place. So that supercoiling of the single strand on one of the ends of the, so unwinding, see this is unwinding, supercoiling takes place here. So that supercoiling also should be stabilized and that tension should be released. It is released by the next enzyme, topoisomerase. So after that, next is the third enzyme is topoisomerase. So topoisomerase is the enzyme in eukaryotes, and DNA gyrase is the enzyme in prokaryotes. Okay. Okay. So this is DNA gyrase. Point number 4, step 4. What happens in the step 4? So primer has to be formed. What is a primer now? So what is the primer for the formation of the second, another strand, new strand? Primer is required. What are these primers? So these primers are nothing but RNA nucleotides. Are you understanding? So these are the primers. So why this primer? What is the need of primer? Anybody? What is the need of primer? Why primer is required? Why is it required? Any one of you? What is the need of primer? Vihan. See, this is how you have to learn. You cannot simply mug up or do. Once you learn like this, you never forget. What is the need of primer? I want to answer from Arya. Arya has made up his mind not to talk only today, I think. I don't know why. Arya, can you please tell me what is the need of primer? Why primer is required, Arya? Arya? Hey guys, you cannot be like this. Hardly few students, and you have to keep talking and tell me what is that, this, everything. Why there is a need for primer? See, the main hero here is DNA polymerase. So the DNA polymerase is the hero which can add up the nucleotide and make the polymer. And this hero cannot add up nucleotides only to the chain of nucleotide that is already paired with the parent strand. So what it has to do? So this DNA polymerase cannot link to the first nucleotide. It cannot link to the first nucleotide in a So it can link to the primer. So what happens before DNA polymerase? It's like this. I'm just giving you an association here. So now VIP is there, chief guest. So let's say this is the chief guest VIP for a particular celebration. So he has to go on the dais. Can he walk alone? No. Who walks? Who escorts that person? So is escorted by some other people, some other member belonging to that group, whoever is hosting the celebration. So that is an RNA primase. So RNA primase forms RNA primer. Okay, so it's what is that RNA primer? It's about 10 RNA nucleotides, which is complementary to the parent DNA. So RNA primase comes to the picture here first. So why is there need of for primer? This is your CBSE hot question. Write down the need for a primer. Why? So you have to write it as the main enzyme DNA polymerase. In that DNA polymerase 3 is the main enzyme. I'll come to that. So that DNA polymerase 3 can add nucleotide. So only to the chain of nucleotide that is already paired. Okay, already paired with the parent strand. So therefore, RNA polymerase, which is also called as primase, add up RNA primer. So this is about 10 RNA nucleotide complementary to the parent strand. And this DNA polymerase now recognize that primer and next it continues the next nucleotide. So already it is there. So it is escorting. So now the hero starts putting the nucleotide and later on. So now it starts putting the nucleotide like this. This is how it acts. Is that clear? So therefore, there is a need for primer. So this is a very, very, very important question. Always they ask. Hot question, I can say. So where this primer is formed? At the free 3' end of one of the strand, fork end of the second strand, primer is formed. Is that clear? All of you. So next enzyme. Enzyme number. Let me shift this here. Okay. So number 4, enzyme. RNA for the RNA primer, primase. So primase is the enzyme for RNA primer. So point number 5 now. What is next point? So now the separate strand is separated. So this separated strands, wherever replication fork now functional. So as a nitrogenous base, here I am showing you. So now let's say that here it is replication is, so let's say 3', 5', here it is 5', 3'. So now where and how the replication occurs? Yeah, anybody? Anybody, can you tell me how the replication occurs? So for the formation of new chain, to start the new chain, RNA polymerase is there, or primase is there. So now, how is the synthesis of RNA primer takes place? 1 mark question. So what do you write? Arya, how is the synthesis of RNA polymerase takes place? So from what is the direction of the formation of RNA polymerase? Yes. Arya, I am waiting for you. I'm waiting for your answer. What is the direction of the synthesis of RNA primase? So RNA primase, what is the synthesis? Direction of synthesis of DNA, new DNA strand. This is an important question and children make wrong in this particular question. So what is the synthesis of new DNA strand? Anybody? Helicase over, single stranded binding, proteins over, then RNA primase over. So now RNA primase, from where to where it joins? 5' to 3' or 3' to 5'? That's my question.
From 3' to 5'.
RNA primase, synthesis of RNA primase takes place in 5' to 3' direction. Please understand. So now RNA primer is added and it is added from 5' to 3' it occurs. Is that clear? Are you understanding? Is that clear? So it is from 5' to 3'. Keep this in mind. So now I have here, let me draw this fork once again properly. So this is what you need to draw. So 5' to 3' and 3' to 5'. Okay. So from where to where now? RNA primase. So from where to where? Quickly. Nobody is answering. How? Amrita. So is this okay? Is this the upper strand or lower strand? Where it joins? Where the RNA primase joins? Guys, 5' to 3'. It joins 5' to 3' direction. It takes place. So now what happens? What is the next step? How 5' to 3' means which direction, students? So this is here, it is this direction, 5' to 3', right? So, and here in this, so it has to come from this direction. So if it has to come from this direction, so then how is it? So, okay, I'm sorry, I have to draw in the fork. How? So it is 5' to 3'. So it goes like this. and here it goes like this. So now it can continuously go here. Whereas here, if it goes like this, it ends. So then what is the next process then? What is it? So the formation of new chain takes place in 5' to 3'. Always listen, always remember, sorry. So this is the thing. So as a result, one chain of DNA is continuously formed, and that's called as leading strand, and the other DNA. So this is leading strand. The formation of the another DNA so begins from the center actually, and not from the terminal point, because not from the terminal point of the opened fork. So therefore this chain is discontinuous. So it has to stop here. It has to stop here. Now this will open, this will open, this will open. So it has to stop, and it has to keep forming that in fragments. So these fragments are called as Okazaki fragments. These fragments are called as Okazaki fragments. So it's named after Reggie Okazaki, Japanese scientist. Is that clear? So it's named after Reggie Okazaki, R-E-I-J-I, Reggie Okazaki. So these fragments, each of them will have around 1,000 to 2,000 base pairs in prokaryotes and 100 to 200 base pairs in eukaryotes, because eukaryotes open up in multiple, it has multiple replicons, we can say like that. So every time RNA polymerase is required. So RNA polymerase bind here, and now this forms. So RNA polymerase primer is bound here. RNA polymerase is required for forming RNA. Sorry, RNA primase is required for RNA primer. So now, how these fragments are finally joined? Finally, these fragments, so these bits and pieces are joined by DNA ligase. So this is the next enzyme. Now you have to count all the enzymes and go on telling one of you. So next enzyme is DNA ligase. Ligase means glue, gluing together, DNA ligase. It's named by Khorana. Okay, and because this strand has bits and pieces, this strand is called lagging strand. So one is the leading strand, which is a continuous, and another is discontinuous, and replication is said to be semi-discontinuous. Okay, so leading strand, once again, leading strand, replicating strand, which gives a continuously replication happens without gap. Lagging strand is a replication strand where the DNA is formed in short segments or fragments, and it's discontinuous. Okay, and leading strand does not require DNA ligase, whereas lagging strand require DNA ligase for the gluing of this of the joining these fragments. So the direction of growth in leading strand, what is the direction of growth in leading strand? What is the direction of growth? Direction of growth is 5' to 3' continuous. Here, how is here? Is it 3' to 5'? Each fragment is 5' to 3'. Of course, that is how. Okay, each fragment is 5' to 3 dash. Here it is continuous 5' to 3'. Now, what is the final step now? Last step. Once all these are formed, so now proofreading takes place. So after you write the book, what you do? You proofread everything. So what is that proofreading? So now suppose somewhere there is wrong spelling, wrong grammar. So then you rectified. Similarly, a wrong base is rectified. So here the very important point is what? Here, the formation of the new chain is catalyzed by DNA polymerase. Very, very important and the hero of the complete process. So in prokaryotes, so this is an important hot point. So in prokaryotes, so this DNA polymerase are of 3 types. Which are they? Anybody? Which are they? In prokaryotes, this DNA polymerase of 3 types. Have you ever understood that, heard it? Anyone in your textbook, is it given? It is DNA polymerase 1. I have written all these. So DNA polymerase 2 and DNA polymerase 3. So let me show you the notes and teach you so that we can do it a little faster instead of writing. Okay, so this is, yeah. Yeah, so in eukaryotes, so there are 5 and prokaryotes, there are 3. There are 3 in number. Oh, it got disconnected 1 second. Yeah, look here. So this DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So now DNA polymerase 3 is the main enzyme. DNA polymerase 3 is the main enzyme. Okay, so it's about 10 different types of peptide chains and called dimer. So it is a replicase. Is that clear? So this is the main enzyme. So in eukaryotes, this is an extra point, so little higher point. So there are 5 different types of DNA polymerase, so which is on the lagging strand. So that is alpha, beta, gamma, delta, and epsilon. Epsilon helps in proofreading in eukaryotes. Epsilon, this is. Okay, so DNA polymerase is discovered by Delucia and Keynes. Delucia and Keynes discovered this DNA polymerase. Is this much clear to all of you? Is it clear? All of you, is it clear? See, this is ORI. This is how it happens. So removal of DNA or primer. So now primer has to be removed. Primer has no job there. So only to activate DNA polymerase 3, it is there. So it is done by DNA polymerase 1. So then joining the fragments by DNA ligase. So in eukaryotes, each individual zone of chromosomes replicates as a discrete section, replication units in eukaryotes. So which is 1 million base pairs. So this is an important point. So after DNA replication, any failure in cell division, suppose the cell does not divide, what will happen? Anybody? So now the cell, so now the replication, so in this, so in S phase of the cell cycle, DNA replication occurs. So chromosome will have sister chromatids. So after the replication, suppose cell division is not happening, what it results in? This is your CBSE question, one more question, hot question. What it results in? This is a fantastic question, common sense. You need not study that. Arya? After replication of DNA, if there is no cytokinesis, it results in dash. MCQ. Aneuploidy, euploidy, polyploidy, haploidy. What is your answer? What's your answer? Aneuploidy, euploidy, polyploidy, haploidy. What should be your answer? Are you there? Students, are you there? All of you are there. Amrita, Arya, Vihan. Amrita, tell me what it results in. Any failure in cell division after DNA replication results in aneuploidy. This is NEET question. Aneuploidy, polyploidy, haploidy, euploidy. Polyploidy, all these you have learned in the previous lesson. It results in what? Array, one student, at least I am expecting that answer. Arya, Vihan, Amrita, at least one of you. Are you people so dumb? No, I don't think. You're intelligent students only. Cell does not divide. Cytokinesis would not occur. What happens, Arya, if cytokinesis would not occur? So DNA is doubled. Everything happens, but cytokinesis not happens. So then there will be double the amount of DNA chromosomes and it results in polyploidy. Simple. Okay, so now we have here so many enzymes. Come on, I want one of you fatafat list all the enzymes, whatever I told you. Quickly, enzyme function. I want Vihan to do it. Vihan, over to Vihan. Vihan, beta, please quickly do it, dear. Vihan, I'm expecting you to do it, dear. Oh my God, this is one. Vihan, tell me. Children, you have to learn this lesson in class, maximum. Which are the enzymes we listed now? This is what is the funda of this complete replication. Amrita, am I audible? Can you please message me? Am I audible? Am I audible? Yes, thank you. So Vihan, come on. Vihan. I'm going to help you, Vihan. If you try it out, you learn better. And this is for your NCRT exam, CBSE also, it's required, not alone for this. Come on, Vihan. Okay, so very first, I'll tell you. Unwinding has to happen, DNA. So which is the enzyme? Unwinding has to happen. Which is the enzyme? Students, shall we wind up the class then? Do you want me wind up the class? I'm helping you out, no? One by one, come on, go on giving me the answer like how we do a recipe. That way I'm teaching you this particular concept. Very first, the parent strand has to be unwind. Which enzyme? Helicase. Amrita, please unmute and answer. That's okay, dear. Please okay, Arya is not there. Helicase. So next, so the soon after unwinding. So the cofactor for helicase is, okay. So the cofactor for helicase is, please do write, this is your NEET point, that is magnesium acts as a cofactor for unwinding for helicase. To swivel, to unwind, anything, any of such sort of questions would be asked. Okay, so next, soon after that, so then it has to get stabilized. Which enzyme? Unwinding, stabilization of the unwound DNA strand. Which enzyme? Stabilization. Quickly. Stabilizing, single-stranded binding protein. Single-stranded binding protein. Next, supercoiling occurs with the end of the single strand. So that supercoiling, the release, the tension, which enzyme release the tension due to that supercoiling in the single strand? Which enzyme? See, third enzyme, which is that third enzyme? Topoisomerase and DNA gyrase in prokaryotes. That is topoisomerase. Okay. So soon after that, primer is required. So I also told you the need for primer. So primer, RNA primer, which is about 100 nucleotides. So which enzyme will add the nucleotides? Which enzyme add the nucleotides, RNA nucleotides? Quickly. Which enzyme is that? Soon after that, nucleotides has to get added. It's by RNA polymerase or DNA dependent RNA polymerase. Okay, so or primase. If you forget, primase. So 4 to 12 nucleotides, around 10 nucleotides long. You can write it like that. So this is 5' to 3' end. So soon after that, so what is the next enzyme? So now comes the hero, that is DNA polymerase 3. DNA polymerase 3 helps in adding the further nucleotides and form the 5' to 3' end. One strand continuous and the other strand discontinuous. So therefore, it is semi-discontinuous. So where these nucleotides come from? These nucleotides come from nucleoplasm after activation, I told you. So now comes the DNA ligase. So Okazaki fragments are connected by DNA ligase. Is that clear? So now proofreading. So proofreading is done by DNA polymerase 1. DNA polymerase 1. So therefore, that main enzyme is DNA polymerase 3. Proofreading is done by DNA polymerase 1. So what is the action of DNA polymerase 2? It's the least active enzyme. So it helps in repairing of DNA if there is DNA polymerase 1 absent. Only it comes into picture when DNA polymerase 1 or 3 absent, it will take up the role of these two. So this much is all about replication. Let's understand a little bit about RNA before going to transcription today. So after RNA, I'll give you a break and then we can go with transcription fast. So RNA world, let's understand that. Anish, can you please tell me whether am I audible and can you see the screen? Please let me know. Anish, am I audible? Oh, Anish is not there, sorry. Vihan, am I audible? Vihan, am I audible? Amrita? Amrita, how much ever you have volume is, please unmute and tell me, am I audible, dear? So RNA is structurally same as DNA, but there are some differences. So what is that difference? The nitrogen base in RNA is uracil in the place of thymine. So instead of thymine is not there. In the place of thymine, it is uracil. So that's one thing you need to keep in mind. Uracil, purine, adenine, guanine, pyrimidine, cytosine and thymine in DNA, uracil and cytosine in RNA. So point number 2, RNA has ribose sugar. DNA has deoxy in the second place. RNA has ribose sugar. So RNA is made up of one polypeptide chain. That means it's a single strand, one polypeptide chain. So what are the different types of RNA So how this RNA is found first? So RNA is first discovered in Rio virus. So it is double-stranded. So that means it has two polypeptide chain. So there are two types of RNA. One is genetic RNA or genomic RNA, and another is non-genetic RNA. Okay, so genetic RNA is discovered by Frankel Conrad. And non-genetic RNA is made up of, is basically three: rRNA, tRNA, and mRNA. Okay, rRNA, tRNA, mRNA. Is that clear? Which are those genetic RNA? That is, it's a genetic material only in viruses, some viruses. So example, HIV, COVID, Rio. Okay, so all these are RNA as a genetic material. Such viruses are retroviruses. Why? You will come to know when I teach you transcription. Retroviruses. So non-genetic material, rRNA, tRNA, mRNA. What is rRNA? So rRNA is everywhere. Ribosomal RNA is everywhere. It's about 80% in the cell. It's discovered by Kunz. Discovered by Kunz. So it's found in ribosomes. It's found in, it's produced. Where is it produced? Because it is produced, found in ribosomes. Where do you think it is produced? It's produced in nucleolus. And it is most stable. So they are present in ATS type, okay, so ATS type, they are present in ATS type of ribosomes. Are you understanding? Are you understanding, all of you? And 70S type of ribosomes. So what are the units of ATS type of ribosomes? The units of ATS type are 60 and 40. 70 are 50 and 30. Is that clear? Is it clear? Is that clear, students? Guys, only both of you. Is that clear? We can wind up early also today, but I want you people answer me. Something you should be telling me, no? Yes, ma'am. No, ma'am. You're not able to see the screen? Oh, not able to see the screen, is it? Guys, so please understand, ATS has Amrita and Guys, you have to answer. Otherwise, I will be go on teaching and I would not even come to know whether you are able to hear, able to understand. Only two of you are there, even Arya left. I don't know why. So, Vihan, am I audible? Can you see the screen? Just message me right now. Yes or no, you message me. Were you able to see the screen? Now I have stopped sharing. Were you able to see the screen? Amrita, how about you? Leave that. That's okay. Now I will teach you fatafat fast. I want to go. Okay. So today I want to teach you transcription translation fully. That's not a big thing. That's why. Okay. So fine. So let me share the screen. Okay. So 80S type and 70S type. 80S type in eukaryotes and 70S type in chloroplast, in plastids, in mitochondria and in bacteria. Prokaryotes, which means. Okay, in 60S, there are three types of rRNA. So that are 5S, 5S, 5.8S, and 28S. 40S has only one type, that is 18S. And 50S, so in this, so 80S has totally four types, that means. So prokaryote 70S type, and 70S have two subunits. So the smaller subunit 30, larger subunit 50. 50S has two types of rRNA, 5S, 23S. 5S, 23S. 30S has only one type, that is 16S. So this is what you need to know completely. So 70S has three types, 80S has four types. This is what is the correct answer which you need to choose. So what is the function of this rRNA? So during the protein synthesis, rRNA provides the attachment site to tRNA and messenger RNA attaches them on the ribosomes. So it also helps the transfer RNA, tRNA, to the larger subunit of the ribosome and messenger RNA to the smaller subunit. So tRNA, the larger subunit to the tRNA and smaller subunit to the messenger RNA, it helps that. That is their major work. So now tRNA. So what is this tRNA? So let me take the tRNA picture so you will understand. So instead of drawing, I don't know whether it is given in your NCRT. Is it given in your NCRT tRNA? Quickly tell me. Quickly let me know. Is it given? Array, one of you please unmute and let me know, students. Okay, so let's understand transfer RNA. Transfer RNA has a clover leaf structure. So this is a clover leaf structure. So this is only 10 to 15%. So it's about 10 to 15% of that is present in the cell. So it's synthesized in the nucleus. So this is also known as soluble RNA. It's also known as soluble RNA. sRNA. So this is also called as adapter RNA. It's also called as adapter RNA. So it is the smallest RNA. This is the smallest RNA. Okay, why all these names for it? It's a soluble RNA, it's an adapter RNA, it's the smallest RNA. Because during protein synthesis, its job is to carry the amino acid, act as a carrier of the amino acid or carry amino acid. So it's most complicated structure. Why? So this structure is presented by Hole. Hole presented this clover leaf structure. So this appear clover leaf like, but in three-dimensional structure by Kim. So it appears as inverted L shape. It appears as inverted L shape structure. So there are three nucleotides. See, it is the actually it is single strand. Single strand is folded like this. Okay, single strand is folded like this. So that's why we have this hydrogen bonds in between. Okay, so there are about 3 nucleotides in a particular sequence from 3' to 5'. So all the at that 5' end. So this is having guanosine. So you can see here. So anticodon loop here. So this is the anticodon loop. This one, and this is the acceptor where the amino acid comes and attaches during translation. Okay, so amino acid acceptor. So amino acid binds to this 3'. This is an important point. Please keep in mind. So what is there here? COOH group. So it binds to that group. So these are folded. Some complementary nitrogen bases come together and form the hydrogen bonds, and that remains makes this tRNA folded like this. So there are some places where the hydrogen bond is not there, and they form the loop. Different variable loops are there. Okay. So in these loops, there are abnormal nitrogen bases. That's why abnormal nitrogen bases. Remember, that's why it's not folded, it forms just a loop. Is that clear? So this is one loop, anticodon loop. This is variable loop, another loop. Okay. So, and this is another loop. So this is attachment loop. So anticodon loop, attachment loop are the two major loop here, and DHU loop. Anticodon here down. This is attachment. T loop means attachment loop. And another one is DHU loop. Amino acyl synthetase recognition loop, DHU loop. So what is that actually? So this loop is to recognize the specific amino acyl synthetase enzyme. You can write it down. DHU loop. So loops are those points in the tRNA where there are no hydrogen bonds because of the abnormal nitrogen bases. So therefore, these have specific function during protein synthesis. Are you understanding? So tRNA recognizes its place on mRNA with the help of anticodon. Anticodon helps in recognizes its place, where to go and sit on tmRNA. That's recognition loop or anticodon loop. So attachment loop is another loop. This connects tRNA to the larger subunit of ribosome. So it attaches to the larger subunit, and DHU loop is the loop which recognizes amino acyl synthetase enzyme. That is amino acyl synthetase enzyme. So this tRNA is 10 to 15%. So the last is messenger RNA. This is about 1 to 5% in the cell. And messenger RNA is the name given by Jacob and Monod. Jacob and Monod. So what is this? Where is this genetic messenger RNA is produced? So this is produced by genetic DNA. Formation of the messenger RNA itself is transcription. So why this messenger RNA is required to get transcribed? What is the necessity of this transcription? How is it formed? So that is all we give in the central dogma. So central dogma of protein synthesis. So why there is a need for protein formation? So that is what we shall learn once you come back from your break. So take a break, come sharply at 7:15. We shall continue with what is the need for protein synthesis, and that is transcription and translation. Okay, take a break, both of you, and come back. Okay, so now the question is, why this protein synthesis has to happen? That's the question. Why do you think the protein synthesis has to happen? Why? See, anything, everything is because of gene, we know. A gene is responsible for the different characters in an organism. Okay, so what is that character? That character is the feature, physiological, morphological, or anatomical character. Okay, anatomical means arrangement of anatomical, physiological, or morphological aspects. Now, these characters may have traits. So traits may be 2 traits or many. So that is more than 2 traits or many. So anything. So these are called as alleles. So now, this gene, this concept of alleles is because of how these traits. So suppose if there is a problem in this, so then it leads to mutation. So this, to understand this, let's go back to British physician Archibald Gerard. So he's called as father of biochemical genetics. So he gave a first hint that genes operate through enzymes. Okay, he's the first person. What is that? He did. So he studied lots of inherited human disorders, and they found that inborn errors of metabolism or failure of metabolic machinery in the organism. So Gerard studied alkaptonuria, which is a genetic disorder. What is that? Alkaptonuria, black urine disease. The moment the person passes the urine, it turns black. So why? Why is this? So through the pedigree analysis, as we discussed, the pedigree analysis, he found that, so it's a cause by the inheritance of a pair of recessive gene. So that is alkapton or homogentisic acid. So this acid, why it produces? This is formed during metabolism of two amino acids, that is phenylalanine and tyrosine. So when phenylalanine and tyrosine, two amino acids, so they, in the presence of alkapton oxidase, will be degraded to carbon dioxide and water. So when this alkapton is not there, so it results in alkaptonuria. Urine will have this alkapton, which would get so the person, so the moment alkapton, that urine is passed, it turns black. So because of the not having alkapton oxidase. Okay, so this we can write it as phenylalanine plus tyrosine form alkapton oxidase to form carbon dioxide and water. So later on in 1948, so another two scientists, so they also worked on this neurospora. So they've worked on this neurospora and they found out so that so the prototype, how X-rays form the mutation, X-rays will, how these X-rays would damage the enzymes, so the gene, so gene mutation happens and therefore it results in different aspects. So this is the point, this is all extra we have told. So this is by Beadle and Totem. Okay, Neurospora crassa. So nutritional mutants they obtained. So they called it as auxotrophs. So that means which is not able to produce their own metabolite here, auxotroph, not able to produce its own metabolite. So why all these? Because of not having proper enzyme. So that means this enzyme, how is it possible for the formation of enzyme? You know that all enzymes are proteinaceous in nature. So this Beadle and Totem gave this as one gene, one enzyme hypothesis. Today, we know that one gene, one enzyme. So enzyme means what? Enzyme is a polypeptide. So today we call it as one gene, one polypeptide hypothesis, which is altered form of Beadle and Totem's hypothesis, and it's given by Yanofsky. Are you understanding? So this is one gene, one polypeptide. So one gene has a functional unit which is called cistron. Okay, so that's what we have understood. So this complete picture, okay, this complete picture. So what is this cistron? Cistron is a segment of DNA which has a proper base pairs that code for one polypeptide chain. A ribosomal molecule or performs any specific function. Now, this central dogma, this complete picture is associated with central dogma, which is given by Temin and Baltimore. So what is there according to this dogma, this hypothesis? So a gene, so formation of transcription, so formation of RNA, messenger over DNA. So that is the first form of gene expression. A gene is expressed. So that is how the gene is expressed in the form of through enzyme. So how this enzyme is formed? Because of a particular gene. So this they told it as prokaryotes, that is DNA forms messenger RNA and which we call it as transcription, and this messenger RNA will form will get translated to a particular polypeptide chain. So in prokaryotes, this messenger RNA is a polycistronic. In eukaryotic, it is monocistronic. Polycistronic. In eukaryotes, it is monocistronic. So what is this transcription? Let us understand now. So let me teach that with some pictures so that it is much more clearer and you understand better. Okay, Amrita? Amrita? Yeah, only you are there. Shall we continue? I want to continue because let them watch the, probably they might watch the video. So let me continue. Shall we continue, dear? Okay, good. So if you are leaving, you please let me know. There's no point in teaching them, or else I can record and send the video also. That's the reason. Anyway, we can do this and okay. So this is central dogma of molecular biology. So let's understand this transcription. What is transcription? Transcription is the copying genetic material from one strand of DNA into RNA. So here adenine, A always pairs with P in DNA. Because it is RNA, A always pairs with uracil instead of thymine. This is one important point you need to remember. So this transcription is always mediated by RNA polymerase. Their DNA polymerase, here it is RNA polymerase. Are you understanding? So here it is RNA polymerase. Where it happens in the nucleus. This is another important point. It happens in the nucleus. Okay. So the strand of DNA with the polarity 3' to 5' always directs the synthesis of RNA and is called as template strand. The other DNA strand, see DNA is a double helix here. So the one with 3' to 5' always acts as a template strand. And that strand is also called as antisense strand. Antisense. Because it forms RNA. It has, that is antisense. The other strand, which is 5' to 3', act as coding strand and called sense strand, because genetic code that's present in this strand is similar to the genetic code present in messenger RNA, except uracil there and thymine here. Is that clear? So that's called as sense strand or coding strand. That means the coding strand of DNA has the same sequence or same nitrogenous bases as the RNA which is transcribed. So except thymine and uracil. This is the meaning. Now, how this transcription occurs, to understand how the transcription occurs. So we need to know the transcription unit. So why both strands are not copied during transcription? CBSE question. Why do you think both are not transcribed? Because the code for protein is different in both the strand. So this would complicate the formation of amino acid or polypeptide chain, that is protein. If two RNA molecules are produced simultaneously, that would be complementary each other and form a double strand RNA and prevent translation. These are the 2 points. So please do write these 2 points. Why both strands are not copied? Only the strand 3' to 5' acts as a template. That means RNA synthesis, mRNA synthesis happens in 5' to 3' as the same as replication. Please remember the formation of mRNA is 5' to 3'. Is that clear? This is very important. So, transcription unit. To understand transcription, we need to know the transcription unit. So, what is the transcription unit? It's a segment of DNA between the sites of initiation and termination. So, there are 2 sites, initiation site and the terminating sites. So, these are the 2 units. What are the units they have? Promoter, structural gene, that is, and terminator. So what is the promoter? It's a region, though this is 3' to 5' coding strand, and this this is coding strand. So this is non-coding, coding. So this is antisense, this is sense. So these are the 2 DNA strands. Now, so what is this promoter to begin with? What's this promoter? So promoter is actually a region on DNA molecule. So here to this promoter, RNA polymerase binds to. RNA polymerase bind to this promoter and initiates the transcription. Where this promoter is present on the DNA, that's an important point. Always to the left. It's present always to the left of structural DNA genes. And towards the 5' end of the coding strand and 3' end of the template strand. So this is a template strand and this is a coding strand. To the 3' end of the coding template strand, promoter is present. In eukaryotes, this is an important point. The promoter has adenine thymine rich region, and that region is called TATA region, that is thymine adenine, thymine adenine, which is called as TATA. So that box is called as TATA box. Okay, so where is this TATA box? So this TATA box is 25 nucleotides in the upstream of this, and this TATA box is discovered by Goldberg-Hargnes. So it's also called as Goldberg-Hargnes box. Okay, I think I have mentioned that here somewhere. Okay, here, upstream of the gene, it is present. Okay, Goldberg-Hargnes bond. So in prokaryotes, this is called as Pribnow box. Please remember, in prokaryotes, it is Pribnow box. In eukaryotes, it is Goldberg-Hargnes, named after Hargnes. So along with promoter, eukaryotes always require even enhancer. So eukaryotes requires enhancer along with promoter. This is another point. May note it down. Enhancer. Now, the other end of this has terminator. What is the terminator here? Terminate means, as the name says, it stops. It's a downstream. Upstream this side and downstream. Downstream is always to the right. Okay, and it's towards the 5' of template and 3 dash of coding. Okay, so that's terminate, that stops transcription. And what are these structural genes in between? So in the area of this template strand, that which transcribes or which are involved in transcription or formation of RNA are structural genes. So structural genes may be monocistronic or polycistronic. So monocistronic means which carry one. Have I written that? No. So please write down. I have written here in structural genes may be monocistronic. So the meaning of monocistronic is they carry the genes for single polypeptide. Okay, so one polypeptide chain, and polycistronic genes carry information for the synthesis of more than one polypeptide. One polypeptide. So what is this gene? It's a functional unit of inheritance. So it's very difficult to literally define a gene with respect to DNA sequence. Okay, so cistron is the functional unit. So you can underline here, cistron is the functional unit. Okay, so of DNA coding for polypeptide during protein synthesis. During oh, sorry, it got deleted. Are you able to see the screen? Amrita? Are you able to see the screen? Okay. So this is about the transcription unit. So transcription unit plus 25 nucleotide upstream, which is which is a Tata box in eukaryotes and Pribnow box in prokaryotes. Okay, so this much is the information about the transcription unit. So now, how this transcription unit happens? So about this structural units, so monocistronic, it is seen in eukaryotes, and here coding sequence, that is exons, express sequence are interrupted by introns. Exons are coding sequence, introns are interrupting, not coding for anything. Okay, so exons appear in messenger RNA, introns do not. Okay, so this is how it happens. Let me show you here. Okay, transcription unit. So how this happens, transcription? The process of transcription happens in 3 steps. Okay, transcription in prokaryotes happens in cytoplasm because there is no nucleus. Whereas in eukaryotes, it happens in the nucleus. And it happens with the help of an enzyme, DNA-dependent RNA polymerase. DNA-dependent RNA polymerase. So this catalyzes the transcription of all the 3 types of RNAs, messenger RNA, tRNA, rRNA. So all these 3 are necessary for protein, synthesizing a protein. So in prokaryotes, it's polycistronic and continuous. That means polycistronic means many polypeptide chains formed at once, and it's continuous. The moment a messenger RNA is forming, polypeptide chain enzymes are also forming and coming. It will, because it happens in the cytoplasm. Whereas in eukaryotes, it's monocistronics. That means one cistron responsible for one polypeptide. So many such cistrons has to be produced. So now this takes place in 3 stages: initiation, elongation, termination. What is initiation? So this is nothing but RNA polymerase once. See, let's understand the initiation program. So initiation here. RNA polymerase binds the enzyme. So at the promoter. So promoter is the beginning region. So here RNA polymerase binds at the promoter site. So the moment it binds, unwinding of the double helix takes place. So now the initiation begins. Initiation begins with the factor, Rho factor. What's that factor called as? Rho factor. So this is present in RNA polymerase, initiates the RNA synthesis. Rho factor. Is that clear? So this is initiation, RNA polymerase. So this uses nucleoside triphosphate as substrate and polymerases in the template dependent fashion. So this initiates that. So always remember RNA polymerase, okay, binds to the promoter site. Local unwinding of DNA takes place. And initiation factor is this one, Rho or sigma factor. This is also called as Rho, present in the RNA polymerase. This is present in RNA polymerase, initiates the synthesis. Okay, so point number 2, elongation. So now, this RNA synthesis takes place as the same way as DNA replication, that is 5' to 3'. So all these activated ribonucleotide triphosphate, that is ATP, GTP, UTP, CTP, are on getting added, polymerization. So, and this is always complementary to the template strand and the DNA sequence. So this happens in the second step. Now, once it's formed, it will come to the termination. And here in the termination, this is another factor. So termination happens. So this is a Rho factor. I'm sorry, that is sigma. This is Rho. This is sigma. Sigma is written like this. And Rho is written downwards like this. So this Rho factor stops here. Termination factor stops the synthesis of this RNA chain. It separates RNA polymerase as well as the newly formed DNA. Sorry. This separates. Rho factor stops the synthesis of this chain. It separates RNA polymerase enzyme, newly formed RNA strand also. So this terminates. So now the processing of this is required. So what is that processing? So this point is very, very, very important here, what I have shown here. So at the end of a gene, there are stop sequence. The simplet stop, what is that stop signal? Is a series of guanine, cytosine, base pairs, followed by a series of adenine, thymine, base pairs. So the RNA transcript of this stop region forms a GC, that is hairpin, followed by 4 or more. This is what? See, it forms a GC hairpin. That is loop, hairpin loop. I have missed out this. followed by 4 or more uracil ribonucleotides, as you see here. So this hairpin causes the RNA polymerase to pause. Okay, so this is another way of termination immediately after the polymerase has synthesized it. So now the pairing of uracil with DNA, that is adenine and uracil in the RNA instead of T. So the weakest of the 4 hybrid base pairs. Okay, so instead the RNA strand dissociates from the DNA within the transcription bubble and transcription stops. This is how it happens. Okay, so this is the detailed information. So how this transcription happens in prokaryotes, that's in bacteria. So transcription and translation happens together. Why? Because it happens in the cytoplasm. There is no nucleus. So soon after the messenger RNA, so that is RNA is produced. So it's a polycistronic. So messenger RNA go on forming many polynucleotide, and translation also takes place within the same compartment, because that happens in cytoplasm, both. Okay, so that is what you need to understand here. Whereas in eukaryotes, it's monocistronic, and here there are certain points you need to note down. So here it is very complex structure. There are 3 RNA polymerases. The primary transcript, that is heteronuclear RNA. So this HNRNA. So what is that? Because they contain, why do you call it heteronuclear? Because they have introns, exons, introns, exons. So what are these introns and exons? So what are these introns and exons? Why do you call them introns and exons? What are introns and exons? As I told you. So what are introns and exons? Exons are functional, introns are non-functional. So hence, introns have to be removed from everywhere. So that is why we call it heteronuclear RNA. Okay, so there are 3 RNA polymerases in eukaryotes. RNA polymerase 1 transcribes rRNA. RNA polymerase 2 transcribes heterogeneous nuclear RNA, and RNA polymerase 3 transcribes small nuclear RNA, snRNA. These are the different rRNA. It is formed. This is a high end. Just understand RNA polymerase. Okay. So now, how this RNA, messenger RNA comes out from it? So how it happens? What is that? Capping is required. Why this capping is required? So as this, I told you about this introns and exons. So these introns and exons first discovered by Sharp and Roberts. Sharp and Roberts. So they are also called as, so these non-functional part along with the functional part. So that's why we call them as split genes also. They're also called as split genes, split personality, both, having both. Okay, so that is non-functional part intron and functional part exon. So this transcription, that is in eukaryotes, transcription produces split genes in, and these split genes contain both coding and non-coding sequence and called as heterogeneous nuclear RNA. And they are very unstable. So that's why they require methyl guanosine to be added. So addition of this methyl guanosine, okay, inversion of this methyl guanosine is added and cap like it is. So this is called as capping. So now they come out methyl guanosine is added. So this is about 200 to 300 nucleotide of adenalic is added. So it is adenalic acid are added to the 3' end. Please remember that is added to this. Okay, so this is polyadenylation. So here many in the tail. So that is 3' end tail. So 5' end is the head. So adenalic acid is added, which is called as capping. And nucleotides of, sorry, methylene, at this, once again, sorry, I'm repeating here. What is capping? Capping is addition of methyl guanosine. Capping is the addition of methyl guanosine here. This is methyl guanosine. Here you can see. And 200 to 300 nucleotides of 200 to 300 nucleotides of adenalic acid is added to the other end. The front end always we call it as head. That's why we call it as capping. The other end is called as tail. So that's why we call it as tail. And this is called as adding many adenalic acid is called polyadenylation. So once this capping and tailing is done, so this messenger RNA becomes stable. Is that clear? Is that clear? So this is how it is done. So now this is the proper stable messenger RNA. So these are all not required for you. Okay, poly A tail. So I have written here in detail. So that's all not required. So now splicing. The next is splicing. So removal of introns. Introns are intervening sequences of RNA which do not code for the proteins are called introns. So they are removed from the primary transcript in the nucleus only. Okay, so once they are removed, so these fragments, exons, are together connected with the help of ligase. Okay, so they are ligated to form proper messenger RNA only with exons. Now it comes out from the nucleus to the cytoplasm. So this molecular machine that accomplishes the task of splicing is spliceosome. This enzyme is called as spliceosome. So smaller nuclear RNA molecules that recognize splice site is a major sequence here. You need to know. So is there non-split genes? Yes. Prokaryotic genes are non-split. So non-split, that means not having split personality. Non-split genes are prokaryotic genes, prokaryotic RNA. Okay, so this much is the information about transcription. So once that is promoter, structural gene coding, and terminator, sigma rho. Okay, so this much is the information. Once it terminates, so the messenger RNA, it is capping, guanosine capping, and 200 to 300 polyadenylation tail. Now it becomes stable. So now it comes out. So before coming out, spliceosome axonate to form the split the splice the introns, and again they are ligated together to form a sensible messenger RNA is ready. So this is called as transcription in eukaryotes. Arya, whenever you feel like you're joining. Oh, screen wasn't visible. Oh my God. Let me show you the screen. See, here, what is that we learned? So I taught you transcription. Please take a look. Once again, I'm going to repeat transcription here. So genetic information copied from one strand of DNA to another to form an RNA is transcription. So what is the difference with respect to replication? Here, adenine pairs with uracil instead of thymine in RNA. So what is the direction? 5' to 3'. So therefore, the one which is with 3' to 5' act as a template. And it becomes antisense because it does not code, it does not seem the same that of messenger RNA. So it's called as antisense. The one which is 3' to 5' is a template because direction of formation is 5' to 3'. The one which is 5' to 3' is the same as good as the messenger RNA. So therefore it's a coding strand. So why both the transcription, both the strands are not copied? So this is an important point I told you, because the code for protein is different in both the strands. So this will complicate translation. That means formation of protein. Point number 2. So is 2 RNA molecules are produced simultaneously, what would happen? It will give rise to the complication. So therefore it forms a double-stranded RNA and prevents translation. So this much is the point. So what is transcription? It's a segment of DNA between the site of initiation and termination. So it consists of 3 regions: promoter, structure, structural gene, and terminator. What is promoter? The one which is located here at the upstream here. So that is, so binding site for RNA polymerase, which is located at the upstream. And these are the structural genes where transcription takes place. And this is the terminator. Okay. So what is transcription? So it has promoter, structural gene, and terminator. So now, in prokaryotes, this is polycistronic, because many polypeptide chains are formed at once because it happens in the cytoplasm only. In eukaryotes, it's monocistronic. So here, so each is formed by, because of one polypeptide chain is formed at a time. And it is interrupted by introns, which are non-functional. So that's why we call them as split genes. That's what I told you. Is that clear? So this enzyme is DNA-dependent RNA polymerase. So this enzyme binds to the box, Tata box, that is the upstream, wherever T, that is TA, lots of TA is present, and hence we call it as Tata box. So that's what it's called as Tata box, and is located 25 nucleotide upstream, whereas similar is present in the prokaryotic DNA, that is 10 sequence away. So this is in the upstream, and this is called as Pribnow box. Tata box eukaryotes, Pribnow box prokaryotes. So now RNA synthesized soon after that. So in prokaryotes, so the first step is elongation. Elongation is addition of ribonucleotides one by one, and termination. It forms the chain, that is elongation, that is initiation by sigma factor, and elongation, addition of polynucleotides, termination by rho factor takes place. So once the termination takes place, so termination forms because of the loop sometimes, and loop cannot transcribe, so then it stops. Okay. So in bacteria, termination, after termination, immediately both as and when. So you can see in this picture, as and when the transcription is taking place, even the protein formation. See here, amino acid chain is forming at the same time. So here one amino acid, here one amino acid, here one amino acid. That's what is happening. So that's why we call it as polycistronic here. And it happens in the cytoplasm simultaneously. Both translation, the moment translation as transcription is happening, translation is will also take place continuously. So that's why we say it is coupled together. So whereas in eukaryotes, it's a complex process because the split genes have to be spliced. So introns should be removed and the exons should be ligated together. Okay, so here each one of them are responsible for this one. So for this activation or the processing is required. This processing happens because of capping and tailing. Capping is the addition of the 7-guanosine, methylene guanosine, here M-guanosine. So at the heading region, so which is called as capping, and at the tail region, it is addition of 200 to 300 adenylate acid, which is called as polyadenylation. So once this is added, the messenger RNA becomes much more stable and functional. So now this comes out to the cytoplasm. So once it comes out to the cytoplasm, translation begins. So how this translation begins? So why this translation begins? To understand the translation, we need to understand certain concept that is all about amino acid. So you know that the chain of amino acid itself is, what is the chain of amino acid? Itself is a protein. Polypeptide chain we call it as. So this polypeptide chain code for a particular enzyme, which is a protein. So how this happens? So that itself is translation. Before going to translation, it's very much important to understand genetic code. So which is given by, this genetic code is given by, term given by George Gamow. So George Gamow is the person who gave the term genetics. So what is this genetic code? It's a relation between the sequence of amino acids in a polypeptide chain and nucleotide sequence of DNA. Sequence of amino acid in the polypeptide chains and DNA. So there are 20 amino acid which participates in protein synthesis. Okay, so there are 20 amino acids. Oh, this zooming only. Okay. See, there occur 20 types of amino acids that participate in protein synthesis. So that means DNA contains information of the synthesis of this any polypeptide with any sequence of this amino acid, which is sensible. So in transcription, what happened? Information is transferred from DNA to messenger RNA, isn't it? In the form of a complementary nitrogen base pairing happened. So now this messenger RNA contain code for each amino acid, and these codes are called as, each code is called codon. So therefore, what is a codon? It's a nucleotide sequence on messenger RNA which code for a particular amino acid. And this code is the genetic code sequence of the messenger Okay, I'm sorry. So the moment there is another class, I'm getting this disturbance. It's getting cut. So let me figure out why it happens. Okay, the sequence of 3 base pair determining a single amino acid is a codon. So how this codon came to the light? What are the different historical background? Let's see. So George Gamow, this person, so he suggested that for coding 20 amino acid, the code should be made up of 3 nucleotides. So actually, they worked for many nucleotide permutation combination, and then they figured out 64 codons. So that is 4 to the power of 3. So which is 64 codons. So what is the contribution of different scientists here? You need to know. Hargobind Khorana developed the chemical method in synthesizing RNA molecule. Okay, so this person is the Hargobind Khorana. So he developed the chemical method in synthesizing RNA molecule. So with a homopolymer and copolymer, Marshall Nirenberg developed cell-free system for protein synthesis, cell-free system, and Severo Okawa. So that is polynucleotide phosphorylase is used to polymerize RNA with defined sequence in a template independent manner. What do you understand by this? So what happens if the genetic code is siglet instead of 4 to the power of, so if it is 4, so then why not like that? Why it should be 4 to the power of 3? All these questions you need to answer now. So why codon is a combination of 3 nitrogen bases? If the genetic code is in singlet, that is, if the codon is a combination of only 1 nitrogen base, so that is, if it is in singlet, so A, C, G, U. So then what happens? So this is insufficient. So 4 codons we get. Insufficient for 20 types of amino acid. Isn't it? If it is 4 to the power of 2, what would happen? Okay, so these are the 20 amino acids. So please understand this particular point. So there are 20 amino acid. If it is 1, so then only 4. If it is 4 to the power of 2, 16 codons. And even that is insufficient. We have 20 here. So that's why 4 to the power of 3, which is 64 codons, and some are repetitive. So that's why Gamow pointed out in 1954, not very recent also, I mean, not very old also. So this is the dictionary of polypeptide chain. So this is how it is. So the chemical method, what do you mean by chemical method developed by Hargobind Khorana? So this became an instrument in synthesizing RNA molecule. So that means different combination of bases. They are mixed. So Marshall Nirenberg also helped in synthesizing, finally helped the code to be deciphered. Okay. So what are the characteristic or the salient features? Very, very, very favorite question of CBSE of genetic code. Codon is triplet. So they have 61 codon for amino acid, but there are 64 in the list. So what are those 3 codons? 3 codons do not code for anything, and they are called as stop codons. Please keep this in mind and do not forget. So these are UAA, UAG, UGA. Which are the stop codons? Very, very important. Stop codons are UAA, UAG, and UGA. UAA called as ochre, UAG called as amber, and UGA called as opal. Okay. AUG has dual functions. So it code for methionine and act as initiator codon. And methionine is the first amino acid and formyl methionine in prokaryotes. So this point you need to remember. So amino acid has many codons except AUG. This point all of you remember, except AUG for methionine and UGG for tryptophan. Okay, so that means degeneracy of genetic code. What do you mean by that? There are 64 codons for 20 amino acid. So most of the amino acid except these two, methionine and tryptophan. Are you understanding? Methionine and tryptophan can be coded by more than one codon. All of them have many codons except methionine and tryptophan. This is called as degeneracy of genetic code, degenerate codes. And this is discovered by Braunfeld and Nirenberg. This is discovered by Braunfeld, F-I-E-L-D, Braunfeld and Nirenberg. So remember this, AUG, methionine, UGG, tryptophan. So what are the other different codons? Here you can see, leucine has so many, CUU, here, leucine has CUA, CUG, UAA, you can see here leucine. So leucine has these two. Serine, where do you find serine? Serine is here, serine is here. Okay, serine has, which are those? Here, UCU, UCC, UCA, UCG, AGC, and AGU. This is all serine. Arginine has so many, CGU, CGC, arginine. Okay, so here it is there, arginine, and one more, so arginine, aspargine, here, lysine. Leucine is different, lysine is different. That means everything has more than one, except methionine. So what is methionine? What is the methionine? As I told you, quickly tell me. AUG for methionine, which is the start codon. And what is the tryptophan? What's the tryptophan? UGG for tryptophan. Is that clear? Is that clear, students? Suppose genes and DNA are best understood by mutation studies. So how do we know genes and DNA proper? Because of mutation, because of disorders, all these. So any deletion or rearrangement in DNA would occur in loss or gain of a gene, which we learned in the previous chapter. So therefore, insertion of deletion of one or two base changes the reading frame from the point of insertion. And this is called as frame shift mutation, which we learned in the previous chapter. So suppose AUG, UAG, all these are there. Insertion of one base pair here will result in change in the reading frame. So this is called as frame shift mutation. Okay, so insertion of 3 base pair, reading frame is unaltered. Okay, so suppose if you add 3 or its multiple bases, so more than one multiple codon, the reading frame remains unaltered from the point onwards. Hence, one or multiple amino acids are inserted or deleted. So that proves triplet. So how do you prove that genetic code is triplet? So by adding 1 or 2 nitrogenous base, so then reading frame changes and you get a different polypeptide, which is a defective enzyme, and therefore the gene expression changes, which results in mutation or disorder. Suppose when you add 3 multiple bases, so then it results in a proper base reading, even though here also it's altered. So one or multiple amino acids are inserted or deleted. So therefore, we can say that this proves that codon is triplet. That means you can still read. Okay, so this much is we did all about RNA. So I taught you all about RNA. So in the next class, let's understand translation. And for translation, I will show you a beautiful video. Oh, again. Since when you people are not able to see the screen, guys? Okay. So I told all about. Since when, Amrita? Could you see the genetic code fully? Amrita, could you see the genetic code fully? Okay, so I'll just do the genetic code and continue with translation in the next class. Do not worry. Okay, so we can wind up the class today. And in the next class, I'll take up translation and we will learn in detail all about the translation. All right. So thank you, all of you. Okay, okay, I understood.
Thank you,
I
ma'am.
don't know. So next class I would be in the here. Okay. Thank you. Oh, Arya, now you are speaking great. Only for thank you, you are so active, full of life, Arya.
No, ma'am, I was listening. I was just not well, so.
You're not well. Your voice doesn't sound like that. It sounds all right.
I was not well for a few days.
Okay, not an issue, but I would appreciate if you participate in the, if you interact. Okay.
Yes, ma'am.
Okay. Good night.
Good night, ma'am. Have a nice day.
Thank you.